It was aptly entitled Cures in the Cupboard, Sharing and Repurposing Assets, which I think in this era of diminishing resources for drug discovery and development, especially for innovation of the process, this panel brings some really interesting perspectives to this important topic area. The bios for all of them, I should introduce myself first. I'm Howard Sewell, the Chief Science Officer and Executive Vice President of the Prostate Cancer Foundation. I'm also a Senior Fellow of the Milken Institute. And we have a really terrific group of diverse people on our panel today. You can read their bios. Barry Bunin, to my left, is the CEO of the Collaborative Drug Discovery Company. Kate Marusa is the Manager and Research Facilitation. Oh, wrong way. Kate? Icebreaker. There you go. Is the Manager and Research of Facilitation Industry Alliances Clinical and Translational Science Center at UC Davis. Melinda Moree is the CEO of BioVentures for Global Health. And Josh Summers, the Executive Director of the Chordoma Foundation. So welcome to the panel and welcome to you too. So I want to start out with a story about a drug in a cupboard that is actually going to be reviewed by the FDA and likely to be approved in the first half of this year. It's called Abiraterone. And Abiraterone, which is now owned by Johnson & Johnson, was a little fuzzy white powder sitting on the shelf in a cupboard at the Royal Marsden Cancer Institute in London. And a very wise urologist from Los Angeles found the compound, thought it was just another sort of Me Too drug to be used for the treatment of prostate cancer. Well, it turns out that the mechanism of action was very unique. They built a company, prosecuted a phase 3 clinical trial, along the way raised a lot of money, took the company public, and within 4 years' time, it's a positive phase 3 trial for advanced prostate cancer and is likely to be approved. So that's a success story. But I would say in general, this is an area of great heavy lifting. And one of the thoughts is, it sort of brings into a couple of different notions to mind. Repurposing existing drugs that are already approved by the FDA that might be lost in a merger and acquisition, or perhaps doesn't quite make the market headline that a pharmaceutical company would want. And then really the discovery of new drugs from chemical libraries of FDA-approved drugs for new indications. And we're going to be getting into both of these. So let me start with the question, do we really believe that there are a lot of these compounds that are laying around? And Kate, maybe you can bring us up to date on what you're doing in this area, because the way you're looking at FDA-approved drugs is pretty, it's critical and it's novel, and you should share that with this group.
Right. So for the past 2 years, I chair a project which is called CTSA Pharmaceutical Assets Portal, and maybe some of you came to the presentation yesterday. The goal of the project is to match academic scientists with those shelved drugs in the cupboards. And I want to make a disclaimer that the drugs that we're looking for are not yet FDA approved, but have been discontinued in their clinical development at the phase 1 or phase 2 due to a variety of reasons, as Howard pointed out. It could be because the company got acquired. Big Pharma reorganizes every month, and so a lot of projects get stopped for no apparent reason to all of us outsiders. The drugs may not have met efficacy endpoints or safety endpoints. So for whatever reasons those are, they are not being currently developed, and some of those passed their patent expiration date. And I want to say some kudos to Pfizer Indications Discovery Group. And Dean, if you can wave your hand. So Pfizer Indications Discovery has been extremely supportive in this effort, and their goals to externalize their shelf compound portfolio closely matched the goals of the NIH and specifically some of the CTSA programs in order to make that match. Mm-hmm. So how many are out there? I think there are different counts, and there are probably thousands and thousands of those compounds. But the questions really are whether the compound that you find in the cupboard is really adequate for the new indication. And in our 2-year work with Pfizer and our experience in that, we have to say that it's very difficult to make the match between the new indication and the compound that currently exists. So it's not that trivial. Mm-hmm. And a little bit later on, I'll go into some more of details.
Okay. Now, Barry, you bring a different perspective to this. What is your, how are you repurposing? How are you finding these drugs in the cupboard?
Well, I think harkening back to what Kate said, there's both compounds that are approved drugs, and then there are a lot of compounds which are in the clinic which have shown either safety or efficacy in humans, which still have had a large investment in them. In CDD, we have a platform for sharing data selectively and securely, and we also have a set of the known drugs from Chris Lipinski and their therapeutic indications. So there have been estimates of the number of small molecules that you could even build are around 10 to the 60th, like larger than the number of universes in the molecules in the universe. And there's only a few thousand drugs. So there is something special about the compounds that have gotten through all these hoops. And so it's interesting. And what else is interesting is there's starting to be a growing awareness in some of the harder areas like Alzheimer's, or where a pharma may have a phase 2 compound that is safe, but they don't quite know the right patient population, or they want someone else to work on it. So I think there is a movement towards drug repurposing, and for good reason.
Now, you're a chemist.
Yeah,
thank you.
Got my periodic table tie.
He has a very cool tie, very nice tie. But you mentioned the name Chris Lipinski,
right?
And that's an important name in the area of drug discovery and development. And for those in the audience that may not know who this person is, because it really adds a layer of rigor to what you're doing. You should explain.
Right. So what Chris did, it's kind of an obvious thing to do, but while he was at Pfizer, he looked at all the compounds that failed in the clinic and those that succeeded. He was able to get a simple rule. It's called Lipinski Rule of 5, so very easy to remember, where you could just statistically look and see with 80% accuracy which compounds would succeed or fail. So if the molecular weight was above 500, too large, it would get digested in the stomach. If the water solubility wasn't good enough, it wouldn't go through the blood. And so you could a priori have a sense of this. And so it's a proxy, and there's exceptions with natural products and infectious diseases. But in general, you can't really argue with it because it's just a statistical analysis. So for example, in the CDD software, we're profiling all the molecules with this Lipinski Rule of 5 so researchers can know up front. And part of what we're looking to do in the pre-competitive space is start to use other ADME and tox filters against molecules with pharma as they start to engage in new ways. But the last thing I'll say about Chris Lipinski, he provided our first public data set, and since then from Johns Hopkins we have extended that to some of the clinical compounds that Kate spoke about. But one of our case studies on this drug repurposing was to look at compounds to use in combination to overcome the resistance to malaria. And we were able to find some known drugs that actually would do this in human red blood cells, looking through the list that Lipinski provided there, and also some new compounds from researchers at UCSF. So this is an example where you can have different people, some with the assays, some with the compounds, some with the hypothesis, and get them to work together in a more collaborative way.
It's very interesting. So once in a while, disease strikes close to home, and you can either take it laying down, when it's a rare disease especially, or you can stand up and do something about it. So we have a really special story here today in Josh, and with his mother, who who have started a foundation to deal with a rare disease that involves heroism and deep intellectual diving, and we want to hear all about what you guys are doing because it's very important.
Well, thank you. So I just, in brief, I was diagnosed with a very rare form of bone cancer back in 2006 when I was an undergrad at Duke University. The type of cancer is called chordoma. It can occur anywhere along the spine. Mine just happened to be in the very center of my head. But I think the important piece of information to consider is not necessarily the specifics of chordoma, but more that chordoma is one of just hundreds of rare cancers, which together actually make up a pretty substantial fraction of all cancers. But when I was diagnosed, I ended up, through some serendipity, landing in a lab at Duke, which just happened to be the only federally funded lab in the country that was studying chordoma. And started really getting into it and making some progress and learning a lot. But we realized pretty quickly that the work that was going on in this one individual lab was not going to lead to a cure in my lifetime, or really in time to save any of the patients who needed cures quickly. And so we really started thinking broader and very strategically about how do you, as quickly as possible, find potential treatments for this disease. And we pulled together a great scientific advisory board, and we've had international research workshops. One of the centerpieces of the roadmap that we've put together is just trying to see, I mean, it's almost common sense to see whether there's anything that's out there on the shelf or potentially that's been shelved that could potentially be beneficial for chordoma patients. So I think your first question for Kate was, do we think there are things out there that are shelved? Well, I think it would be incredibly presumptuous to assume that we have selected the only disease that a particular drug may be efficacious for. We know that there is an incredible amount of shared biology among different cancers, among different diseases. We know that there are off-target effects of lots of different drugs. So to assume that there was just one or two indications that a particular drug would work for just seems a little bit presumptuous. And so the approach we've taken is really two-pronged. The first is, all right, let's see if we can very rationally identify the biological underpinning of chordoma, which we've made significant progress on, and we've now identified what we think is a very, very good target, or several good targets actually, and kind of in a forward manner, rationally develop compounds to interact with those targets. Or we could just say what's available right now that could help patients that are in desperate need today. And to answer that second question, we've worked with the NIH Chemical Genomics Center to screen all of the approved drugs. And so that's where we are right now. We've got a number of hits, but it turns out that it's quite difficult to go from, all right, we've got some compounds that are active among 2,800 in cell lines to, all right, how do we figure out why they're active and which ones we take into clinical trials, which ones are most important.
So how's it going?
Very, very, very well. Better than I ever possibly could have imagined, actually. We've been able to really bring together an incredible team of researchers to collaborate with us. Certainly, being able to partner with the NIH Chemical Genomics Center has just been a godsend. And so, yeah, we've got 45 compounds that appear to be active. And now there's just a whole host of hurdles. We can, I guess, get into them later. But we've got compounds that are approved that may potentially be active. I mean, these are drugs that patients could start taking tomorrow if they were really promising. And we've also got a series of targets that we think are very, very important. One in particular, which I'm very excited about because it was first identified as being important for cancer in chordoma. It's a gene called brachyury. But since that first paper was published last year showing that it was important for chordoma, there have been 3 or 4 other papers showing that it's also very important for colon cancer, for pancreatic cancer, for lung cancer. And I think there's kind of a growing awareness that this may actually be an important target in cancer generally. So there's now several different groups that have become interested.
Mm-hmm. Fabulous. So you heard about where some of the medicines might be coming from, but there's a world of need out there in the area of global health. And we're fortunate to have one of the world's experts on global health with us today. Melinda, if you could share with us what you're doing, what you're trying to achieve, and where your drugs are in the cupboard for your lofty goal of distributing life-saving and disease-preventing treatments into the third world?
There was in the panel this morning a discussion about the differences or the similarities between these groups who are working in global health and the groups that are working on rare diseases. And I think there was a missed opportunity this morning because it made it sound like these are drastically different models. And really, they're not different models. So I think for those of you who are working on rare diseases, what we're trying to do in global health is really fairly similar. So what are we missing? We're missing tools for drug development, which is what most of the rare diseases are missing as well. We're missing an understanding of the basic biology. It's very difficult to do clinical trials. You have to aggregate the patients. And companies aren't going to do this on their own because we're dealing with little market. In the case of rare diseases, because there are so few people. In the case of global health, because there are massive numbers of people, but they can't really afford to pay, certainly Western prices for their medicines. So I think there's a lot here that's very similar. So when we talk about some of these mechanisms, the idea that we're all kind of working in our, each group tends to work in its own little world, and then each community tends to work in its own little world, that I think we're really missing an opportunity to perhaps do some things together. One difference that I would point out is the patient advocacy groups, which I'm just in awe of the patient advocacy groups in the rare diseases. We talk about this all the time in global health and try to figure out how you get somebody with malaria to come and testify in front of Congress and how many family members they've had that have been touched by malaria or schistosomiasis. Not many. So we're at a real disadvantage when it comes to the advocacy efforts. It does help to have Bill and Melinda Gates, though, out there. So that we do have a few things lined up for us. So a lot of what we try and do, BioVentures for Global Health, is really trying to create more opportunities for industry to be involved in developing drugs, vaccines, and diagnostics for neglected diseases. And there are lots of barriers to that, but a couple that we've identified. One is an information barrier. So I think the kinds of things that we're talking about today first of all, people don't even know what the names of these diseases are. So putting that out there and saying, here's what we mean by neglected tropical diseases. And then starting to go much deeper into the science and figuring out, well, what are the targets for drug development? What are the tools? Who might you partner with for this? I think not dissimilar to the model that many are using for rare diseases. But when it comes to this particular topic of repurposing, part of the reason that I spend a lot of time with Faster Cures is that I think that there is a lot in common here. So when you look at the basic biology, I think Barry said it well. We have a lot of drugs that work against different targets, and a lot of those targets are shared. So we could go through, I have pages here actually from someone on my team much smarter than me in this area, about all the kinds of shared targets between great commercial opportunities and some of the neglected tropical diseases. But we are not able to build on what has been learned because of the proprietary nature of drug development that I think we're starting to move away from, but is still, pretty much how the industry makes its money. So, I'll just leave with a couple of opportunities that I see. Because of these shared targets, I think there's a lot more opportunity for people to share pre-competitive information, and I think our definition of what's pre-competitive is changing. And I think that's a really good thing. And I think that's going to help us all. And then the other thing is figuring out ways to pool some of this information. And I really do not mean to make everything open source and open access. Companies still have to make money. You still have to be able to protect your discoveries. That is the fundamental nature of doing business. But that model is not a robust model right now. So I think there's a lot of opportunity. And one of the things that's interesting about global health is that it's not threatening because there aren't big markets at stake. So so when we talk to people about being able to do work in a different way, a lot of the major companies are talking about sharing data and information in tuberculosis drugs. Because even though a lot of people have tuberculosis, the companies really don't see this as a big market. And so they're more willing and more open to share data in this area. So I think there's a lot of opportunity right now, but I think it's also incumbent on those of us working in the non-industry side to kind of get our act together and to pool things as much as possible. I talk to CEOs who are very happy to be doing this work. Everybody feels good about working on global health, rare diseases. There's no shortage of people and companies who want to work on these. But from a corporate perspective, what the CEOs keep saying is, I get approached every other day from some group who's doing some little piece of something and they want us to collaborate with them. And he's kind of saying there's only so much mind share that you can put toward these things. So I think it is incumbent on us to streamline this a bit and to think about ways that we can work together to get something much greater going.
So it brings to thought, listening to Josh, some of the other notions that there's actually an oncology drug called RAD-001 that's in clinical trials for the treatment of epilepsy. Can you imagine a cancer drug being repurposed as a medication for a seizure disorder? That's because scientists figured out that the pathways that are activated, the signaling pathways in certain cancers, are shared in the epileptic brains of children with uncontrollable seizure disorders. So there's a lot of hope here. Let's talk a little bit about some of the challenges and start out with intellectual property. I mean, we have to face the realities that for a medicine to get developed by a pharmaceutical company or even a small biotech company, it has to have an intellectual property runway. So let's start down here. Tell us how you deal with intellectual property in your libraries.
Maybe we can switch to the slides, and then we can look at the whole universe of barriers that surround drug repurposing.
Is this Barry or this is Barry?
Let's go down.
Tell me when I get to yours.
You will see a big wheel with lots of blobs on it.
The second set, please.
Okay. Well, I'll just start talking while they bring the slides up. The slides that you're about to see will show you different. Pardon me?
They're all falling off.
Sorry.
I'll try not to move. Is it me?
No, it's not you.
Okay. So.
I'll hold it. Thank you.
So the Portal project is the first attempt to do drug repurposing on the national scale. So we have the partners in the form of Pfizer, who is present here, and National Institutes of Health. We also have members, which is 50 universities across the country. And we have about 500 individual members in the portal. And so issues of IP and the issues of funding and the issues of how to actually put the compounds in the pool and issues of how to match the academics and the assets are all very important issues. So there's absolutely no doubt about that. And if addressing, I don't know if we're going to have slides or not. Maybe not. Uh-oh.
Yours didn't make it in, so just keep going.
That's a bummer. Okay, so I'll just keep going then. So in terms of the issues of IP, there are compounds in the shelved assets that are still under IP protection, and those still have significant value to the companies. And many companies have established drug repurposing units within their own companies to deal with those assets and to figure out what to do. And certainly there's millions and millions of dollars invested in these pipelines. However, there are some assets that are no longer under patent protection or about to lose the patent protection. And I think those are a little bit easier to talk about, and those are the ones that we hope that the pharma companies will be able to put in a shared pool with the various protections that we provide for them so that the IP issue is not as acute.
And Barry, what are you doing with intellectual property in your company?
Yeah, so at Collaborative Drug Discovery, we have web-based software for handling the chemical structures, which correspond to the composition of matter patents, and the bioactivity, which are the utility patents. And when I formed the company, initially I was going to call it open source drug discovery, and that's too radical for this world and the way people want to work. And so more nuanced way to do things, or to give everyone the power and the opportunity to decide if, when, and with whom to work with in which way in terms of intellectual property agreements. So we talk about the technology allows you to collaborate, but you can withhold information if you want. Or you can share biodata without structures, or structures without biodata. So related, we're also funded by the Bill and Melinda Gates Foundation with BioVentures for Global Health. And they're working with Glaxo on developing this pool for open innovation, which it's sort of goodwill gesture, and Glaxo's put a number of patents into it. But it allows people to do what they want. And actually, Bill Gates has been doing this with really rich billionaires to give money to charity, but they decide where they give money to charity. So there's similar themes here. And in our case, we had malaria data from Glaxo, which was private, for 6 months, and then they were willing to share a subset of their data, and Novartis for tuberculosis. With Pfizer, they're not willing to, in their collaboration with us, they're doing a lot of other things with Kate. We have a collaboration with them where they're going to share the models for predicting ADME and tox, and they don't need to share the compounds. So I think new approaches and new technologies can catalyze collaborations and drug repurposing in a way that doesn't compromise IP, or that gives people more flexibility, and then they're more comfortable to do more.
And Melinda, in your quest to solve global health issues, how does intellectual property act as a barrier in your work?
Well, when I started out working for the Malaria Vaccine Initiative, which in the global health world we call these product development partnerships. I'm not sure what your exact term is in the rare disease community. And so much talk about intellectual property impeding, poor people getting access to new medicines. And so I did a fair amount of work, trying to figure out how true is this. And I've come to the conclusion that about 95% of it is kind of permanent employment for people with an ideological bent about intellectual property, and about 5% of it are about real issues. But for us, what we've tried to do, talking about this pool for open innovation, is it's not so much where is IP a barrier, because that's not actually what we're facing. When the right incentives are in place and you ask companies to do things, the record of companies stepping up and cooperating is just phenomenal. I know of all these different product development partnerships on TB drugs, vaccines, malaria drugs, and vaccines. I know of maybe 2 companies where groups have not been able to come to some kind of deal about the assets that they were looking for. So I don't think it serves as a big barrier. But the bigger question, I think, and a much more interesting question, is how can we build on the intellectual property as we go forward? And that, I think, we don't do a very good job of. So there's a tremendous amount of knowledge. There are compound libraries. There are all these assets that sit in pharmaceutical companies that I don't think we've done a great job at figuring out how do you build on what's been learned. And the asset may be access to compounds, but what people really want to know is, what did you learn about those compounds? What did you learn about that compound library? How would you think about that going forward? And that's something that's much harder to have a discussion about, because then you start getting into the data and not just the compounds themselves. And that does really start getting into proprietary space about safety and other things like that. So for me, that's the area for the future, is how do we actually use what people have learned to build on that, instead of, there's just not enough money to recreate what other people have done. There's not enough money out there to do that. And so again, I think the neglected diseases are a good place, because pre-competitive, I think, goes for a very long distance in neglected diseases than what it does in commercial targets. So this pool for open innovation started with GlaxoSmithKline. Alnylam joined. GSK put over 2,000 patents in. Alnylam put their entire patent portfolio in. It's restricted to neglected tropical diseases. And let's just say the barriers to entry are pretty low, because if you make the barriers to entry so high, no one's going to join. And there's quite a bit of discussion now among pharma companies to join in with this and to really make it a much bigger and better resource. And again, the aim is not to get rid of IP as a barrier and not to get rid of IP. is how do you use the knowledge and the information to build on that and just get further faster?
And Josh, you're an army of one, and we have another army of one in the audience, and I just want to acknowledge, which is, my friend Audrey Gordon in the back, whose family has been affected by a rare disease called progeria, and they're repurposing a drug, a farnesyl transferase inhibitor, and other compounds for, how many children in the world?
About 200.
200 total affected by progeria, an accelerated aging disorder, and they're just doing such great work. But I know some of their barriers, they're substantial. They're just, 200 children in the world affected with this disease. You have a rare disease, and I know it's easy to say that you're going to, learn, that drugs, medicines should have multiple indications. We don't always know that, but you have lots of barriers that you're trying to break down. Why don't you pick a couple of them, the ones that really keep you from sleeping well at night, and tell us how you're dealing with that.
Yeah, absolutely. So, the barriers specific to chordoma are many. I would just want to step back for a moment and maybe talk about one barrier that I think is not unique to chordoma. So I think the kind of paradigm that we're all talking about here is that you have some chemical space, some number of compounds that are out there, and some very large number of diseases, so maybe 10,000 diseases, let's say, on the order of that magnitude. And there's a matching process whereby you want to essentially find out if all these trillions and trillions of compounds, or maybe some set of them, are active in your tens of thousands of diseases. And the problem is you have all these different segments. You have some compounds that are in various companies, you have some that are in the public domain, you have some that are approved. But really, especially for a small organization and for any of these diseases that are not massive, you don't really have the capacity to go to all these different sources. You can't go and create relationships with every company. The transaction costs are just way too high. And so I do think that pooling these compounds, getting them all in one place and having some common mechanism whereby you can start screening lots of different diseases against the broadest possible library of compounds is going to be the most efficient way for everyone, not just for rare cancers like chordoma. And so there was a speaker here named Artie Rai. She's a law professor at Duke, and she's come up with just a really a brilliant kind of legal framework for how one might go about getting compounds both under intellectual property protection and not into a common pool and enabling a sort of en masse screening process against these compounds. So I'd encourage everyone to check the paper out. It's called Navigating Pathways Across the Valley of Death. And so I'm trying to proselytize and tell everyone I possibly can about this paper because it seems like it could solve a lot of the problems that we're all talking about. Just really quickly, specific to chordoma and also to other rare cancers, one of the biggest problems we have is simply models. So in order to find out if there's a compound that is potentially active, you have to have models and you have to have validated models. And in the case of chordoma, we found that there were a number of cell lines out in the literature that we have found to be invalid. And this is a rampant problem across all types of cancer. It's estimated that about 30% of all cancer cell lines, and therefore presumably 30% of the data that's generated with these cell lines are just invalid. They're not what people think they are. So I think this is a huge overlooked problem that people kind of poo-poo, but it's really, could you imagine, putting drugs into patients based on data that was generated in things that are garbage, essentially. And so in the case of chordoma, we've invalidated 5 cell lines. We have validated 2 cell lines, and the Chordoma Foundation has obtained the right about 2 years ago to distribute one of those cell lines. And just this month, we obtained the right to distribute the second one. We have about 28 researchers that we've sent the one cell line to that we expect to send the second one to very quickly. And then one of the barriers that we're running up against presently is that we've been fortunate in that there's several companies that have become interested in chordoma and want to screen their compound libraries against these cell lines, or that want to use them for some sort of research and development effort. Well, the academics that own these cell lines want royalties, and the companies view what they're doing for chordoma as not a moneymaker. It's very much a favor. It's a charity project. And so to erect a barrier to say that the academic's going to demand money out of the pharmaceutical company would just stop the project in its tracks. And so we've kind of come in, and I think this is a really important function that a foundation can serve, is appease the academic investigator, say, all right, we'll give you some money if you give your reagent to the company. And in this particular case, we were able to actually negotiate a material transfer agreement such that the foundation has the right to distribute the cell line to companies. So we don't have to have all these bilateral negotiations between universities and the companies and other investigators for that matter can just come to us, and it's in our interest to get these things out there as broadly and as quickly as possible, and we're not going to let it kind of sit on someone's desk for months. So I think that's a real opportunity for across diseases to develop new models, to validate new models, and to kind of untie or unencumber these models from all the universities that are trying to hold on to them, get them in sort of some centralized place so they can be applied to or screened against these pools of compounds. So I guess in summary, it's two focuses, two things that need to happen. You need to get a set of models that are able to be used, and you need to get a set of compounds that are able to be used and just match. And there's lots of permutations there, and I think it can only be done kind of in a systematic manner. Trying to do it scattershot with creating all these different relationships and transactions just becomes too cumbersome. And I think that's why this process, everyone realizes, is so long and cumbersome.
Can I add to that, please?
Of course.
Now, as I probably have my slides back, I want to address this issue of matching, because in our several years of working with Pfizer, and Pfizer really attempting to match their compounds to their academic interests, we found that this is the largest barrier, is to find the overlap between academic interest, commercial interest, and find where the biology really supports the disease. So can I have my slides, please, now? Is that possible, or is it not going to happen today? Okay.
I'm not sure they got your deck in there.
I think they did now.
Just keep going. It's
see.
after Barry.
Barry has.
We like Barry's slides.
Okay, here. Oh, yes.
You can present my slides if you want.
All right. So what we're discussing right now is all these individual issues that may prevent the matching of the compounds, if you wish, to the science. And this is IP, as we discussed. We haven't touched on funding anything. We haven't talked about funding. Who's going to fund all these repurposing studies? The current NIH pathways to funding are very, very crowded. And I have heard the rumors that even a few of those that are available is going to sunset pretty shortly. So who's going to fund? Who's going to do it? Where are we going to get the compounds? All of these are issues. And I'm just going to show you one that we have identified in the past 2 years as being one of the most problematic, but there is a solution to it. And again, how do you match the compounds to the people? Where are those people who are going to do a quality science that are really going to prove disease biology? And so what we have developed together with Pfizer is a method of identifying those people who are eager, willing, and able to prove the disease biology with the compounds that are out there. So the premise there is that every compound has a target, and then you can group the targets according to a certain diseases that they have been associated with. And then you can further group it with the actual living, breathing investigators that study this target in this particular disease. And so on the overlap of the target, disease, and investigator, you can make a match. And that sounds ridiculously simple, but it required a considerable amount of programmatic effort, because the social networking softwares out there right now, like Alexis and such, that claim to do this, actually mostly looking between relationship between the people rather than relationship of a person to a disease or a target. And just to flash that screen for you, we have a website where you can look at the a demo of the program. But here's the target, which is here in orange, which is grouped by strength of the connection to diseases that this target is implicated in, and you can set the barrier high or low if you're looking for one publication, if you're looking for 1,000 publications on the theme, and then you use the and expand, so you find individuals who have published on this target and this disease. You get list of their publications, and then you specifically look for those key publications that that link the target in the disease. So that's our attempt to find those matches, so to find those researchers who have a biology that you need, and not even touching on the compounds. So
Thank you very much for that. You had a comment, Barry?
Somewhat similar to this, bringing different brains together, different people looking at data different way. So it's all about aggregated learning. And if you think about places where things have evolved very fast, such as the internet, where a lot of brains are getting together and working, it's a similar thing that needs to be done here, because there are going to be surprises. Like thalidomide is a classic example where people with multiple myeloma take it now, and Viagra found in phase 2 the indication. So what we've been doing is coming up with a repository where accumulated knowledge, such as done in the areas of global health or rare diseases like what Josh is working on, and the information, the aggregated knowledge is going to, biology is conserved, and so that aggregated knowledge of which drugs work, having a place to learn from each other and work with each other, that's, I think, the key for it to be not a new project from scratch each time. And that's sort of the whole idea. Yeah.
Absolutely. Global health has huge challenges. What are one or two that keep you from sleeping well at night?
Well, I always sleep well at night, so nothing keeps me from sleeping well at
You're
night.
lucky.
I sleep on planes, I sleep standing up, I'm good on that one. For global health, there are two sides of it, and unfortunately people tend to make them as separate things. There's the delivery part of it, which just has huge challenges. So poor countries, very little infrastructure. But despite that, vaccines still manage to get to somewhere about 70 to 80% of the kids worldwide. So there are rays of hope in all of that. The delivery challenges are numerous, and which include the money to buy new medicines, and so that's one challenge. And then the other side is just we've had a dearth of new tools in global health up until about a decade ago. So most of the therapies that are being used are old. Many of them are toxic. They're things that would probably never get licensed in any way today. And that's kind of what the global health community has, is old toxic cures. And so when you're faced with, well, let's see, do I have a chance of actually dying of the cure, or do I, these are the dilemmas that people are facing every day. So I think it's both creating new tools and delivering them. But I think what's really interesting, and why these aren't separate, although they used to be kind of separate communities, is that what we found is that as you generate new tools, people get excited about finding the money to pay for them. And so with the renaissance going on in vaccines, as those have come along, I remember sitting in a meeting a few years ago, and the Gates Foundation had hired someone to do a vaccine landscape. And we looked at the vaccines for diseases of the developing world, and there were like 10 on the list that were coming through the pipeline. And it was very funny, because I'm thinking, like, this is great. And everybody else in the room was like, oh my God, how are we going to get these things, through the pre-qualification process, and where are we going to find the money, and where are we going to... And it's like, but by having the potential, then what we've seen since that is that all these different systems, so the formation of the Global Alliance for Vaccines and Immunizations, which is a great big purchase fund for vaccines, that wouldn't have come about unless people would have felt the pressure that there were actually products to buy and that could save lives. So I think that there's not this either or. Do you figure out how to deliver existing tools, or do you create new ones? They're incredibly synergistic. And so I think, again, it's by looking at the whole value chain, that instead of just looking at our little piece of it, and I think I'll digress for just a second, but I think that's part of what we encounter when we get out of the industrial setting, is that we tend to just look at the piece of value chain that we play in. So if you do delivery, that's the most important thing. If you do clinical trials, that's the most important thing. If you do discovery, that's the most important thing. And the thing is that none of them are important without the other ones. And so you've got to have enough of each of them to be successful. And industry does this every day, or else they don't get paid and they go out of business. So they have a built-in reason for really looking at this as a value chain. But I think that, and you can see it in a meeting like this, is people kind of have their piece of it. And that's the most important piece, and we're all struggling for funding. And so we end up kind of pitching against each other. So I think there's something for just kind of looking at the value chain, taking that approach and understanding the benefits that others can bring. So I've already said in rare diseases, your advocacy, beats our advocacy. And I think there's a lot to learn from that.
Yeah, I certainly agree with that. So I get to give my final vignette for the day, because we only have 5 minutes left, just to say quickly, which should give you some hope. So Gleevec, who I think most of you have heard of, was on the front page, on the cover of Time magazine, heralded as a breakthrough drug in oncology, probably will result in 2, maybe even 3 Nobel Prizes. That was a drug in the cupboard. And it was a drug in the cupboard because it was a compound looking for biology. And once the biology was discovered, they knew when they treated the first 2 patients with chronic myelogenous leukemia that this was a home run. But to give you some hope, my good new friend Josh, gastrointestinal stromal disease is also known to be a target of Gleevec. And yet Novartis said, no, I don't think so. It's small, there aren't that many. Well, fast forward, it's $1 billion indication for Novartis. So all of a sudden, gastrointestinal stromal disease is very important to that company, so don't give up. And then the last thing, I just want to do a quick right through the panels, and then we'll have some time for questions, I believe, correct? This notion of pre-competitive space is important, and we've thought about it in terms of biomarker discovery. How do you get all the stakeholders together, those in not-for-profits, those that have a definite profit motive in corporations? What are the pre-competitive space 1 or 2 issues that you would like to see solved?
When we're talking drugs, we're talking competitive. I don't see how that could be translated. Maybe I'm not understanding the question.
How about a model? How about a biomarker that might affect everybody? I mean, Josh, maybe you've thought about this too, or you've actually brought some of these together.
Yeah, I guess I would say I know a number of the pharmas have developed something called the Pistoia Alliance, and people were having wine in the city of Pistoia, and they decided we're reinventing the wheel too much. So they are talking about technologies, and I'll get back to the dichotomy mentioned before about funding versus tools, and you ultimately need both, but new tools, new technologies, new innovations, new ways of looking at things, that's the real progress, and the funding will grow over time. But I think in the pre-competitive space, it's about bringing the right data together, like Josh was mentioning, this like global structure-activity relationship table of all diseases against all compounds. Every company can create that, and aggregated together as humanity, we can create that. And so that's sort of the definition of the pre-competitive space. And I think there's so much work, so much work goes on, and so few drugs get developed. So the vast majority of data is failed data on what hasn't worked. And even a failed project, when it gets published as a great success. I found this hit or something. And so I think what we've been trying to do is capture all the data, let people have it private, but then if and when they're willing to share it with others, then allow them to open up. And I think that's sort of how the pre-competitive and competitive can kind of equilibrate.
Maybe I can comment on that, since now I understand what the question is. And Dean Welsh here can comment on it better than I mean, don't hide, please, because that was a really Pfizer-driven idea. So when we started talking about involving academic biology in the drug repositioning, the thought there was, well, why not create an alliance between pharma companies where pharma donate the tool compounds against certain mechanisms, and between all the pharmas out there, we can probably cover 99% of mechanisms out there, and you don't need to reveal necessarily the chemistry of it. It doesn't have to be your clinical compound. It just have to be very specific tool. And by sharing the data of what mechanism works in what disease, we can eliminate that waste that every pharma company goes through. Because you try the mechanism, it fails. You never publish on it. Nobody ever knows about it. The second pharma company sinks another $200 million in doing exactly the same thing. So that kind of pre-competitive space that may be quite beneficial for all the stakeholders involved, and hopefully that may be created at some point of time.
Yeah, I mean, even more basic things like clinical trial design, biomarker discovery. We have found that they actually will, the 900-pound gorillas will come into the room with academic experts and actually try to solve problems, and that's been pretty productive. So I'd like to thank our panelists very much, and I believe we have some time for question and answers. So. Maybe just Audrey, if you'd like to say a few things about your foundation, you can do that if you'd like, because it's so close to what we're doing here.
Thank you. I appreciate that opportunity. Progeria is known as the rapid aging disease, and there is a biological link between the disease process of progeria and the kids die of the same heart disease you and I would, and there is some aging mechanisms that are going on there. So in terms of that cross pollination and what we're doing may be important for others, not just rare diseases that we may see, but also the general population. We're really hoping that that story translates into a very happy ending, like I'm sure Josh wants to for Chordoma. So thank you for that opportunity.
I think of things in terms of buckets, old drugs, old targets, old drugs, new targets, new drugs, new targets. If you look at it that way, you get a matrix that kind of clarifies, how you can think about these things. But there's one biological mechanism which is fundamental, and it's coming to fore now, and that's the field of epigenetics. Because there you're dealing with fundamental translational aspects, and many disease states have a translational component to them, and many of these things are in fact modified by chemicals. And I wonder if any one of the panelists would address, it's not that far out, I mean it is occurring now, and there are approved epigenetics drugs, and they are accomplishing something. Is there space for the old drug, new target concept in any of your thinking?
I would just make one real quick comment, and I think this goes to one of the reasons why it's so important to get these pools of compounds and pools of assays, because you never know what assay is going to be the right one. I mean, we can only use the best assay we have right now, and for Chordoma, that's simply cell kill. It's probably a pretty crude assay, and so we don't want to have to reinvent the wheel, create all these relationships again, and do the whole thing over if we find that, let's say, there's an epigenetic assay that we need to be looking at, or some other assay. So I mean, it could be any number of mechanisms that one would want to test against, and let's not reinvent the wheel for each one.
I would say in oncology, which is the area I know the most about, the chemical biology program from the NIH is being applied at UC San Diego as well as at Johns Hopkins to specific prostate cancer problems. And there actually are some hits that are epigenetic modifiers that are being followed up in preclinical studies as we speak.
Yeah, because it is a fundamental mechanism.
It is, we agree.
And it crosses a whole bunch of disease states, many disease states. So it's almost looking at the thing from the bottom up basis rather than the top down. Disease down, actually you're looking at mechanism up at a chromosomal / cellular level.
Right. Certainly in the case of cancer, where the genome is oftentimes scrambled with fusions and translocations and other genetic anomalies, those are epigenetic phenomenon to be certain. So, other questions? Yes, sir.
I guess it's for Melinda. First of all, I just want to acknowledge your wisdom in highlighting the fact that giving a compound isn't really all that helpful compared to giving the data that goes with it and the reams of data that we literally have around our compounds. So I'm wondering if you have any thoughts on how we might better interact in making those data available in a way that is constructive for both the donor and the recipient?
Yeah, so maybe Barry can answer the specifics, and I'll just talk to the like kumbaya, how do we all work together part of it. So I mean, I think that one way is this pool for open innovation. The idea is not, so it's a little separate than what Barry's doing, but the idea is not to just sort of take all data, put it out there, you know, mix and match, but the idea is just to be willing to make it available. So by, I mean, much as what was mentioned, by creating a structure and a framework that companies agree to ahead of time. And by the way, this is a company initiative, right? So it's not the public sector saying, won't you do this, won't you do this? This was a company initiative to do this. And so then a lot of it is just, it's the information on two sides. It's people in the disease community who care so deeply about that, letting companies know what it is they actually want and need. And I think we've done a poor job of articulating that, and certainly the aggregating it, we're not even close to. So I think that's one part of it, because I don't think companies are going to do that, and nor should we expect them to. So I think really articulating what those needs are, and then being able to have a format for going to companies and saying, you know, here's a group that would like access to this for this purpose. But having it kind of, you know, that's when you get into the transaction costs. And so by trying to create a mechanism, a framework, a structure, so that you don't get into all those transaction costs, I think that's the second important thing. And that means everyone has to let go a little bit. And actually the word trust has to enter in here somewhere for that. So I think that that's a really important first part of it, so that companies are maybe not just putting out there publicly, but they're willing to share at the request of someone, and then maybe even willing to work with others. I mean, I sat at a meeting where all the big pharma were there and some of the small ones on TB drugs, and they're all of them have a program in tuberculosis. And they were kind of sitting there looking, and it was like, nobody had any idea what other people were working on, what kind of data they'd gotten, what had failed, what not, and they're not doing this for commercial purposes. So if you're going to use those hard-earned dollars to do good in the world, you want to make sure they're really well targeted. So I think there's some benefit back to the companies as well in just getting better information from people and to guide their own programs on, you know, how do we use our money in the wisest way possible if we're trying to do good in this sector. So I think there are a couple of benefits. So I think it's information, getting good information to people, and then some kind of structure and framework to make that happen without all the transaction costs, at least in the beginning.
Can I add just a little more detail around that? Just with the Pfizer, well, at CDD, our company is a spin out of Lilly, and we're looking to work with groups like Pfizer that want to selectively share data, because it's all the data around the compound that's key. And some folks in the audience, Alf Bingham and Bernard and Tom, but created new ways of looking at this. And another spin out was InnoCentive, where people can put prizes, and then only the people who have permission to see the result get to see it. So I think figuring out this pre-competitive space at a place like this with faster cures, where there are all these foundations, there are ways where you can get aggregated learning within groups of people working together and also accelerate the process. So playing back to the pool idea and just figuring out how it works, I think this venue is almost the perfect venue for this type of topic.
Thank you. And the last point, Audrey?
I just want to bring this all together on a human level, and thank you all. It's initiatives like this that will really give people like Josh, my nephew Sam, and really thousands, millions of others with neglected or rare or other diseases, save us the time and the money that we simply don't have. So thank you very much.
Thank you.
Thank you. Thank you all.
Thank you, Howard.